Zoology Laboratory lecture series, and we will be gonna talking about patterns of organization in animals. So before that, we would be introduced with the strikingly ordered series of changes in evolution, which is reflected in the body plans of your animal kingdom and also for the protists. So these evolutionary changes in your animal body plan might be likened to a road map through a mountain range. So what is most easily depicted are the starting and the ending points are a few of the attractions along the road. So as you go ahead at the top of the mountain, you will see the diversity and the sophisticated body plans of these animals. Whereas when you go downwards or in the lowland, they are more easily depicted. Okay, and they have the most basic and simple body plans as we go ahead with this discussion. So in the learning outcomes, we will be comparing the body form of animals that are radially symmetrical to body forms of animals that are bilaterally symmetrical. And we also have to differentiate the three forms of triploblastic tissue organization. So what is symmetry? Okay, symmetry is describing how the parts are of an animal is arranged around a point or a certain axis. So when you say the symmetry, okay, your animals, like animals and protists, they are organized into different diverse forms. And within this diversity, we need to know the patterns of organization. How are we going to classify these animals? Um, Thus, the concept of symmetry is a beautiful thing in understanding animal organization. Okay, so we have different animal symmetries. We have asymmetry, we have bilateral symmetry, we have radial symmetry. And the first is asymmetry. So this is the absence, okay, of a central axis or a point. So let's say for example, we have the sponges, okay? Don't have a central pointer axis, so are um, they don't possess a symmetry on their body plan. When we say bilateral symmetry, there is a single plane passing through the upper or the anterior and lower surfaces through a longitudinal axis that divides the animal into its left and right images. So for the vertebrates, we are bilaterally symmetrical animals because when we divide ourselves into the left and right image, you can see the mirror image of the both sides. When we say radial symmetry, these body parts are passing through an oral aboral axis. So it divides the animal into these mirror images, let's say the nidarians, okay? And they can be modified into different arrangement of structures or combinations. We have this biradial symmetry and ctenophorans and some amphizoans, and we have the pentaradial symmetry in the echinoderms. So when you say pentaradial, it has a five symmetry. So these are the echinoderms. Some of them are having this five radial symmetry, or the sea stars and the the company. Okay, so this isn't a sponge. As I said earlier, it displays a cell aggregate organization. So this is a red encrusting sponge, Monocora barbadensis, which shows many are asymmetrical. So this is the free-form shape. Next is the radial symmetry. So this oral aboral axis is their mouth and their anus. Okay, um, they have this single location or point on their body. Okay, and it divides the radially symmetrical animals. This is example on the photo. We have this tube coral polyp, the Tubastrea sp, into equal halves. Certain arrangement of internal structures that modify the internal radial symmetry of the sea anemones. So these are the coral polyp. And for the bilateral symmetry, this is a fox, okay, a vertebrate. The planes in terms of a direction useful in locating the parts of a bilateral animal. So we have how many planes that divides? We have the transverse plane, okay, and we also have this frontal plane and the median plane. And the dorsal, posterior, and ventral. We will discuss that one later. So these are the imaginary planes that cuts the animals into their mirror images, okay, or mirror halves. So now we will learn about terms of direction. The term is written on this table and the description. So for you say aboral or aboral, the end opposite the mouth of a radially symmetrical animal. So they are limited only for a radially symmetrical animal. And oral is the end containing the mouth of a radially symmetrical animal. So when say anterior, it's the head end, okay? Usually it is the organized area of the animal's body is in the head, okay, or the cephalic region. So the head end usually it meets the environment that of a bilateral animal. So when you say posterior, it's on the tail end, okay, or the lower portion of the body of the animal. Caudal, it's towards the tail. When say cephalic, it's towards the head, okay, moving to the head. Distal is away from the point of attachment of a structure or the body. When you say the toes are distal to the knee, so the toes are away to the knee. When you say proximal, it's toward the point of attachment of a structure of the body. So the hip is proximal to the knee. So the hip is the proximal to the knee, so towards the point or towards the knee. Okay. Dorsal, it's the back of the animal. That usually upper surface, okay, of the animal. It's synonymous for posterior for animals that walk upright, just like the humans. And ventral, we have the belly of the animal, so it's found on the belly side, usually the lower surface, and it's synonymous with anterior for animals that walks upright. Inferior, that's below a point of reference, so that's below when you say the mouth is inferior to the nose in humans, that means to say that the mouth is below the nose. Superior, that's above a point of reference. So the point of reference is the neck is above the chest, okay, is or superior to the chest. When you say lateral, it's away from the plane that divides a bilateral image to its mirror images. So away from the plane is away from the middle. When you say towards the middle or towards the plane that divides the animal, we call that one as medial or median. So what are the other patterns of organization? So we have this tissue layers and body cavities. Kasi we are not only considering the body symmetry of the animal, because we have a lot of areas to consider or factors to consider. Okay, so in this section we will try to learn about tissue layers and body cavities. So you know that the tissues are aggregate of cells, and body cavities are being developed during the embryonic development of your animal. So when you say the unicellular level of organization, these animals or organisms that are consisting a single cell, okay, so we display this unicellular level of organization. When you say simple unicellular, okay, we refer them as cytoplasmic to emphasize that all living functions are carried out in the confines of your plasma membrane. That's mean to say unicellular. That doesn't mean to say that unicellular is not simple. Okay, they also must provide for the functions of locomotion, food acquisition, digestion, water and ion regulation, sensory perception, and the reproduction in a single cell. So these are the cytoplasmic level of organization for the single-celled organism. When you say diploblastic organization, we have these cells that are organized into tissues. So we call that one as a twofold, okay, diploblastic. Is the simplest tissue level organization. So we are dividing the body parts into two different layers, two embryonic tissue layers. The first one is the ectoderm. It's outside, okay, outside of the skin, which it gives rise to your epidermis and the outer body layer of your body wall. When you say endoderm, it gives rise to the gastrodermis. That means to say the tissue that lines your gut cavity, the internal organs. And this is a diploblastic body plan. So these diploblastic animals have tissues derived from your ectoderm and the endoderm. Between these two layers is a non-cellular, we call that one the mesoglea. And we have a triploblastic organization. Some animals are triploblastic, that is, their tissues are derived from three embryonic layers. As with the diploblastic animals, ectoderm forms the outer layer of the body wall, and the endoderm lines the gut. And a third embryological layer is sandwiched between the ectoderm and your mesoderm and your endoderm. Sorry, and this layer is what we call as the mesoderm, which means the middle. So if you have heard metaphase, okay, it's MESO, which means in the middle. Okay, this gives rise to your supportive, contractile, and blood cells. So these are your triploblastic body plans. Okay, they are derived again from ectoderm, endoderm, and mesoderm. Letter A is a triploblastic acoelomorph pattern. Letter A. Letter B is a triploblastic pseudocoelomate pattern. So there is a pseudocoelom on the middle of your endoderm and your mesoderm. Note that the absence of mesodermal lining on the gut tract. And lastly, we have the triploblastic coelomate pattern, wherein a mesodermally derived tissues completely surround your coelom, such as the peritoneum and your serosa. So why are they advantageous? Because they provide room for organ development, also to provide more surface area for gas diffusion, nutrients, and waste into and out of the organs. It also provides an area for storage, often act as hydrostatic skeletons, and it's providing the vehicle to eliminate, to remove the waste and reproductive products from the body, and to facilitate increased body size. So in summary, we have animals that are organized around a point, so we call them radially symmetrical animals in an oral aboral axis. If it's a bilaterally symmetrical animal, they are arranged in around in a longitudinal axis, a plane that divides the half mirror image of your animal body. A particular organization often associated with a particular way of life. And protists in animals have composed of cells, and animal bodies are organized into tissue layers. They might be diploblastic, triploblastic, acoelomate tripoblastic, pseudocoelomate, or triploblastic coelomate. Body cavities have advantages that promote increased body size and complexity. So what about higher animal taxonomy? So the taxonomy is a study of classifying the organism itself, or the animal, or any living organism. So this is what higher animal taxonomy is. To compare the protostome and the deuterostome animal groups. So traditionally, the Animalia have been considered monophyletic. So Animalia have a single ancestry. So mono, which means one, okay, single ancestry. Because of the impressive similarities in animal cellular organization, about 0.6 billion years ago, at the beginning of the Cambrian period, okay, an evolutionary explosion occurred that resulted in the origin of all modern phyla, along with the other animals that are now extinct. So the Cambrian period washes off the entirety of the animal kingdom before. So animal taxonomy are based, okay, principally on embryological and morphological evidence, okay, but they are being reinterpreted based on molecular evidence. So principally from what? Our RNA studies or ribosomal RNA studies. Five basal phyla was originated independently from other animal groups. So these are Ctenophora, Porifera, Placozoa, Acoelomorpha, and Cnidaria. These five groups, the Cnidaria and Ctenophora are commonly grouped together as radiata based on the presence of radial symmetry. Okay, like the Cnidarians, okay, the polyp, okay, and the diploblastic organization in both groups. Recent studies suggest that the Ctenophora may in fact be triploblastic, and their apparent radial symmetry is secondary. This independent origin of the five phyla is being reflected in just figure 7.12. So these are the basal phyla, okay, the Lophotrochozoa, the Ecdysozoa, and the Deuterostomia. Okay. The use of this molecular data has altered or changed the zoologist's interpretations of animal taxonomy in this tree. An independent origin of the phyla Ctenophora, Porifera, Placozoa, Acoelomorpha, and Cnidaria on the orange basal phyla. Okay, the members of this phyla containing bilaterally symmetrical animals, which are divided into three groups. The Lophotrochozoa are protostomes that do not molt. That means to say, these animals are not shedding. Okay, they include the annelids, mollusks, and others. The Ecdysozoa, because they perform ecdysis, are the protostomes that are molting. Okay, they include the nematodes, the arthropods, and others, and the Deuterostomia, including the echinoderms, hemichordates, and the chordates. So these are your kingdom Animalia. And protostomes are traditionally including the animals in the phyla Platyhelminthes, Nematoda, and Mollusca. So this example is a mollusk, okay, an octopus. In figure 7.13a in E, it shows the developmental characteristics that unite these phyla. So we have this protostome development and the developmental characteristics between the deuterostome versus a protostome development. Okay, so in letter A, we have this spiral and determinate cleavage. In letter B, a mouth, okay, this one, that forms from an embryonic blastophore. So this one is a blastophore, an embryonic blastophore, and it forms a mouth. As schizocilous coelom formation in letter C, and letter D, we have a trochophore larva. A polycate worm is an example of an adult protostome. Letter E, we have a deuterostome development, which is characterized by letter F, which is found in here, a radial and indeterminate cleavage. So this is what we call as the deuterostome. Okay, and letter G, we have the anus. It's the opposite of the protostome. Anus develops in the regions of your blastophore, and then it forms a letter H, an interior-coelom formation, and members of the phylum Chordata are deuterostomes for letter F. So this is a fish, and this is a good image. Okay, so a difference between the early development of the protostomes versus your deuterostomes. Blastula divisions occur as a spiral cleavage, and this protostome division planes are oriented obliquely to the polar major axis. In deuterostome, the cleavage is radial. Okay, that means to say it has a symmetry. Okay, radial, It has a central axis, and they are parallel or perpendicular to the major polar axis. Parallel, opposing sides. And during gastrulation, the protostome's embryo's mouth is given first by your blastophora, while the anus is formed later. But in the deuterostome, it's the vice versa. It's the opposite one. Anus is given first and then the mouth. So we have here also an image showing the protostomes, which are mollusks, annelids, arthropods, and the deuterostomes, which has 8 cell stage during the spiral and the determinate and radial and indeterminate stages, in the coelom formation and the fate of the blastophores. The other traditional group, or we call the deuterostome, in the animals include the echinodermata, okay, the sea stars, or the sea cucumber, hemichordata, in this example, acorn worm, chordata, and others. So the figures 13fi shows a developmental characteristics that unite these phyla. So again, radial cleavage is occurring when the mitotic spindle is oriented perpendicular to the axis of the zygote and results in embryonic cells directly over one another. So for a summary, I'll give you again that the Animalia is a monophyletic group, meaning to say they have originally on the single ancestry, and there are animal phyla that arose independently of any other group. These are the 5 basal phyla. The bilaterally symmetrical animals share a common ancestry, and protostomate animals share the developmental characteristics such as the spiral determinate cleavage, including the Lophotrochozoan and Ecdysozoan lineages. And lastly, the Deuterostomia share developmental characteristics such as your radial indeterminate cleavage. So this is just a beginning, and I need you to study more on this patterns of organization in order for us to better understand the different phyla of your animal kingdom and to enjoy zoology as a course. So thank you so much for listening, and stay safe.